This one here?
Yes, exactly. And this red one, you want to show.
Oh, okay, super.
Okay.
Thank you. Thank you very much. My name is Andres McAllister. I'm Chief Medical Officer at BioInvent. You might have heard of our biotech company. We're very happy and proud of what we're doing. I'll tell you a little bit exactly what we're exactly doing. So the company has grown tremendously in the past few years. We have now several partnerships in all the clinical programs. Big pharma are looking at what we're doing, and we have been able to secure several agreements with them in order for them to provide drugs to our studies. The company is totally focused on immunotherapy of cancer. And of course, this is, if one could want to name a single most important thing that has happened in cancer in the past 20 years is this question of treating cancer by manipulating the immune system. It's now well established, and for the great majority of cancers, you do have that as an option. Yet the medical need is still very much present. The great majority of patients may have respond and stop responding, or not respond at all. So we're actually addressing that very question, which is really the focus of what we're doing. This is just some historical data in the green here. This was the first drug that was approved in 2010. The second set of drugs approved in 2014. This is anti-CTLA-4. This is anti-PD-1. And when you add those two, you have somewhere around the 50% of the patients that respond. But of course, there is a huge medical need here of patients who do not respond or stop responding. And this is related to how much inflammation there is inside the tumor. So something that has come as a realization in the past years is the importance of what happens inside the tumor. It's very different to what happens outside, in the periphery, in blood, everywhere else. What happens inside the tumor is really very of the essence. And that's what we're looking into, and that's what we're trying to address. This is the technology that we use. It's called FIRST. It has been patented and actually very wide and very nicely protected. Basically, you might have known BioInvent in the past. When it was founded on the basis of having a phage display library, which basically it's a way to produce antibodies. And it's a huge collection in a very small tube, so you can actually use that. And basically what we do is we take samples from the tumor directly, and without doing any manipulation of the tumor, we throw the antibody library on top of that, and by then getting rid of the background, so through some sequential steps, you basically end up with antibodies that recognize something in the tumor. Then we test that in animals and make sure and identify those that have anti-tumoral activity without knowing what they're recognizing. We're looking at after the function first. And then once we do that, we identify the antibodies that have a strong anti-tumoral activity. And once we know that, then we go after the target, identify the target, characterize the antibodies that can have similar activity to that target, and then move that into clinical development. And that's what we've been doing in the past 6 years. And as you see here, we've been able to put several programs into the clinic. If you show this to any large pharma executive, they would be very impressed of this amount of clinical studies in such a short period of time. So I think that speaks very nicely of the success of this platform. And I'll tell you a little bit of what we've been doing. We have, when you do that exercise, of course, you fall into some new targets that some people might have known of, but perhaps not a lot. You identify some that are usually more or less characterized, and of course the first that would be a good control are the ones that everybody knows, etc. And we've found basically all of those kinds. One very interesting target, and probably the one that has had the most important antitumoral activity in animals, is this TNF receptor 2. TNF receptor 2 is TNF is a very well-known protein. It is actually recognized by a receptor called TNF receptor 1 that is present in just about every cell in the body. But there is another receptor, which is called TNF receptor 2, and that receptor turns out to be highly expressed inside the tumor microenvironment by cells that are there present in the tumor, and in particular this type of cell called regulatory T cells, which basically suppress the activity of the immune system. By the way, something that we know today that happens inside the tumor is we know that there is a very suppressive activity. So lots of the immune system, components of the immune system, are telling the immune system, stay put, do not recognize the cancer that is actually growing. So cancer is manipulating the immune system inside the tumor. Here's just some of the companies, but I think the interesting thing is that BioInvent has the first in class. This is the leading product in the world. Other companies are pursuing the same thing, but we are really at the front edge of that technology. This is a schematic, and apologies for the busyness of this, of how busy the slide is, but basically it shows, we started a couple of years ago treating patients at a low dose. We've been up to very high doses, and the drug is, for the very, very well tolerated. We can administer it safely, and now we have moved into phase 2 part of the study, where we're looking at patients with lung cancer, ovarian cancer, melanoma, and T-cell lymphomas. And if there is, we can address the reasons for this during the discussion if you're interested. In the meantime, I told you that we had some basically supply agreements with Merck, the manufacturer of the most successful of all of these drugs, which is anti-PD-1, pembrolizumab, and we're basically combining BI-1808, that's how we call this drug, with pembrolizumab. We are about to finish this escalation phase, and then we will move into similar expansion cohorts, that's what we call that, so groups of patients with the same type of tumor, and we will be treating them either as single agent or in combination with pembrolizumab. And just to show you something that has had us very excited, this is a patient with a rare type, this is actually a Swedish patient with a rare type of disease called GIST. It's a form of sarcoma, gastrointestinal sarcoma, and basically what you're looking here is lesions that the patient had when the patient entered the study. This patient had received 12 previous lines of treatment, and this is a very hard to treat, very unresponsive to treatment disease, to both chemotherapy or other type of therapies. What you see is that the tumor at the beginning of the study started growing, and during that phase, probably the immune system was getting boosted, and then at about day 50, the tumor started decreasing. Each one of these are lesions that this patient has, and basically you see that these lesions have actually disappeared, and these lesions are still in the same trajectory, so decreasing. So this is extremely exciting for a type of disease that is very difficult to treat. In the meantime, we also have another type of drug targeting the same receptor. The antibody here is called 1910. Basically, we are in the same journey. We are about here treating patients with the lower doses. This is an agonist antibody. The biology of this target is very interesting, but so far it's been very well tolerated, very safe, so we have not seen any problems. Eventually, when we get to higher doses, we will move also into expansion cohorts as in the previous situation. And I will tell you a little bit about BI-1206, which was the drug that we started to develop a few years ago. Basically, BI-1206 targets a receptor called Fc gamma receptor 2b. Interestingly, if you look at these are the T cells. These are the ones that you have heard about. This ipilimumab, pembrolizumab, etc. These are the inhibitory receptors targeted by a number of antibodies, and basically the brakes of the immune system that tell those cells do not do what you're supposed to do. When you look at the innate aspect of the immune system, so macrophages and antigen-presenting cells, etc. The interesting thing is that there is also several activating receptors, very much like here, but there is a single inhibitory receptor, so a very potent, but only one receptor. And this is the target of BI-1206, which we are developing in solid tumors and non-Hodgkin lymphoma. Basically, what you have is this is the receptor, this is the antibody, recognizes the receptor, and it basically tells this cell what the cell should be doing. So if it is an inhibitory receptor, it basically tells the cell, do not do what you're supposed to do. So one important aspect of this, and I told you about this notion of the tumor microenvironment, is that when you look inside the tumor microenvironment, as opposed to blood, you see that in blood you have very little expression. In the tumor microenvironment, it is really, really highly expressed. And that is the reason why we think that Fc gamma receptor is so important. And here you have 3 different types of diseases where independent studies have shown that if you have a high expression, your disease is very, very aggressive. This is survival. So you see that when you have high expression of Fc gamma receptor 2b, your prognosis is very poor. The same in follicular lymphoma, and the same in this very difficult to treat diffuse large B-cell lymphoma. This is BI-1206 in non-Hodgkin lymphoma. Basically, what we're doing is we started a few years ago. Now we are in the part B of the study, where we're treating patients with the two antibodies, BI-1206 and rituximab. We're also developing a subcutaneous formulation, but there again, we've seen patients with very nice complete responses that have lasted for a very long time, a few partial responses, etc. So all of this is moving along. And something that is very important, and in the interest of time, basically, I'll tell you shortly, is that we presented this data to AstraZeneca, and basically we're now introducing a new arm in the study where we will give a triplet. Cancer, like infectious diseases, is important to treat it at different places. So this is a very interesting development for us. So we will be starting this within the next few months. Also, a strong rationale to combine it with pembrolizumab. There again, Merck is providing the drug for us, and BI-1206 is being combined with pembrolizumab. And right now we are in this, moving into this part of the study where we're doing, we're treating patients with melanoma, lung cancer, and we have a basket cohort where we treat patients with other malignancies. Okay, so finally, another version of that antibody is called 1607. 1607 has been shown to increase the activity of drugs like HER2, targeting agents such as trastuzumab. Again, CTLA-4, this is very interesting because CTLA-4 ipilimumab is a very difficult to use drug because of the toxicity. And what we have shown in animals, at least, is that when you combine it with this drug 1607, you can actually reduce the dose of ipilimumab. This is really a holy grail when you speak with clinicians, because this would allow to unlock the usage of this drug. Right. So we also have generated a series of partnerships with other companies that are developing these antibodies, and the partners are depicted here. And basically, this will be some of the things that we'll be talking about during the year. It's a very exciting year for us. We have a lot of data coming up with this TNF receptor 2 antagonist, BI-1808, also in combination with Pembro. 1206 in combination with Pembro, 1910, that other anti-TNF receptor 2 single agent. We didn't speak about this, but this is a very interesting drug that is a virus that expresses an anti-CTLA-4 antibody, and there again, by the year end, we should have some very more descriptive, of course, early but interesting data. So thank you so much, and happy to take questions and discussion.
Thank you very much, Anders. We can sit over here. If you have any questions, please raise your hand. And if you take it in Swedish, I will translate. Okay. When I read your interim report, Martin, the CEO of BioInvent, wrote that this might be a breakthrough year for your company. And as you described, it's a lot of 6 or 7 studies ongoing.
Yes.
I mean, if everything is successful, you move to another stage, right? And your company, how much can you carry by yourself?
Yeah, that's a very important question. Of course, so the good thing is that we've been very successful in raising capital in the past few years. So we have a good runway until entering 2026. So that's very nice, and actually allows us to pursue our plans with relative calmness. Of course, the data will talk, but what we're seeing, for instance, with that patient that I showed, is that we're starting to see something that despite the great effort that has been made by the pharma and biotech industry, it has not been seen. So this is basically single agent activity, seeing patients respond to your drug alone. So that, I think what we hope to be able to do is to get partners on board, big pharma, et cetera, that would put a lot more muscle into the development of these things. Because of course, the interesting part of this is that is not only for that type of tumor, but this spreads over a number of different tumor types. And once this becomes obvious, and we believe that this year is going to be a key year for us, a lot of these patients will start. We will continue seeing what we have seen already, and I think that will be a major driver for capturing interest from investors and also from potential pharma partners.
Yeah, but you still need to bring everything a little bit further, right?
Yes.
All the projects.
Yeah, all that you have seen here and our current plans are financed by the funds that we have. So, of course, it doesn't take us to approval, which is a different thing. One of the strategies that we follow here is that you might have seen that in some of these, basically in each one of these programs, we treat some rare diseases, so that a study is probably smaller, is probably difficult in the sense that there may be less patients. But for instance, I mentioned T-cell lymphoma, and that is a very difficult to treat disease, and there is not a lot of things that are being developed. So if you see activity there, it's quite straightforward to move toward approval. And with our program, we have been able to capture the attention of the most important sites, both in Europe and the US. And we're actually running our studies there in order to be able to treat those rare patients with these diseases.
Okay, so you don't see any risks when it comes to recruit patients, for instance?
Well, the good thing is that when you're using this, when you're developing these new targets that are exciting and that show promise, etc., these very interesting sites that capture a lot of those patients, they come along, and that's exactly what we're seeing in our study. So, for instance, I mentioned that G's patient. Now that we have shown that to our investigators, etc., they're actually looking for those patients, bringing them in the study, etc. So we have not had patient recruitment issues in those solid tumor studies.
When you now have 6 or so studies at the same time, is there any of these that you think are of particular interest for somebody who's following you?
I think both the target TNF receptor 2 has the advantage that is you're seeing single agent activity, and that is something that you can put forward. The data speaks for itself, so that is very interesting. Fc gamma receptor 2b is a target that has been shown for years to be of importance. And why is it not being pursued by other companies? Because it turns out that finding an antibody that recognizes very specifically Fc gamma receptor 2b is very difficult, because Fc gamma receptor 2b is 95% identical to an activating receptor. So it's very difficult to find an antibody that will only block Fc gamma receptor 2b. That speaks of the quality of the library at BioInvent, which actually, out of a very large screen, there was only 2 antibodies out of the library that were interesting, and we moved one forward because it had other advantages. But that is really the reason why also we believe that Fc gamma receptor 2b is a very, very interesting target. And it's a target that it lends itself to be used in a number of other combinations, which is, I think, very interesting.
Yeah. As a CMO, I mean, I guess you have clouds on the sky, right? Things that you think of. What kind of risks do you see going forward with all your projects? I mean,
well,
obviously, I mean, it's individual projects, and it takes too much time, I think, to dig into every project. But on a macro view.
Yeah, well, I mean, when you're developing drugs, something that you have to worry about every day is safety of the drugs. You're treating patients, you're treating human beings. It could be my mother or somebody like that. So you have to think about that very seriously and act upon it very seriously. Fortunately, we have been very lucky that the targets, and I think is tied up to this notion of directing the activity to the tumor microenvironment. The drugs that we're developing have been very safe. We haven't seen any, we don't have any reasons for concerns today. So I think that's the major risk is having safety issues, safety concerns, things that would preclude the usage of the drug because of off-target effects, etc.
You mentioned the competition. How would you say in your area what is actually going on outside of BioInvent?
There is with the I happen to spend a lot of my life working in the immunotherapy of cancer, and for many years this was very depressing. Nothing really worked. Until 2010, when the ipilimumab showed activity in a phase 3 study, and it was approved later that year. And 4 years after, anti-PD-1s, anti-PD-L1s were approved. Since then, there's been a huge amount of money thrown to the area because, of course, it's very promising. And I showed you all those receptors. There's been those and many others that have been targeted, etc. And I think the frustration has accumulated in the field that nothing else has really proven to be so interesting. What's happening? A lot of these companies have unfortunately failed to show activity in many of these areas. We still think that there is a lot to be done, and so we feel that that we really have an edge in understanding this biology, these biologies.
Yeah, it was very interesting times. I think we will look forward to all the results in the next year. There's a lot of studies going on. Thank you very much for coming.
My pleasure.
We need to move forward.
Thank you.
Thank you.